Maybe slightly different than it says on the program announcement. So the real person who probably would be the best to give this talk is Andy Sakin, but he has a niece that's getting married and could not make it. So I'm going to try to pretend to be Andy, at least in part. But in our lab, we do a lot of imaging genetic studies, different types of quantitative phenotype studies, and I'm really the imaging part of the imaging genetics. I'm a neuroscientist, so I'm not as skilled in the genetic parts of this. So I adapted my talk to what I know. Given that we're going to be using the ADNI study for most of these genetic analysis, I thought I'd talk a little bit about the different phenotypes that are available, and then what's been done so far, at least selected papers on genetic analysis in ADNI. So many of you may know that ADNI provides numerous phenotypes to evaluate genetic variants that might be associated with disease. It's a longitudinal study, and so many of the phenotypes, although maybe not all, are available both on a cross-sectional and a longitudinal basis. The majority of phenotypes are available starting at baseline and then at various points afterwards, which I'll discuss in a little bit. There are a few types of different types of phenotypes that were actually added later on in the study, so they actually aren't available at baseline, and so you have to sort of pay attention to when these phenotypes were evaluated. So this is a graph, which I get to use my pointer, of the different publications, genetic publications by phenotype. So the gray bars are the number of publications using the ADNI genetic data. As you can tell, it's sort of grown kind of rapidly and then maybe leveled off a little bit. But the different colors will tell you the different types of study phenotypes that they used in the different types of studies. So clinical and cognitive phenotypes are by far the most common. But more recently, with the advent of some of the PET tracers and that being done on the full ADNI sample, as well as some advanced MRI, those have sort of come on sort of later as these had been introduced as part of the study. And what they found in a very busy figure is primarily ApoE, which I could just stop there and then walk off. But if you take ApoE out of the equation, then there's a whole bunch of other genes that have been implicated, and these often are the same sorts of genes that show up in the sort of top 20. So my goals are actually a little bit more on the phenotype side, as I said, to sort of describe the ADNI study and what the sort of outline of the way the study is collected, as well as what types of clinical and cognitive phenotypes there are. And then I'll talk a little more about the endophenotypes of the fluid and the imaging measures, and then describe some of the genomic analyses with ADNI phenotypes to date. So ADNI is broadly divided into 4 different phases. It started in the early 2000s with the ADNI-1, which I think we're going to mostly be using data from at this phase. And then in 2010, there was some additional funding as part of the grant opportunities to enroll, specifically targeting people who were in the early part of the mild cognitive impairment spectrum. And then, happily, ADNI got renewed and rolled into ADNI-2, which started in 2011, and just recently, ADNI-3, again, it's been renewed and is continuing on. So there are participants who've been there from the beginning, who've rolled over from ADNI-1 to go to et cetera, et cetera, et cetera. But they have brought in new participants at each stage. And the participants generally fall into categories of cognitively normal older adults, those with significant memory concerns, which is what ADNI called subjective cognitive decline because of the way they assess it, early MCI, late MCI, and then mild Alzheimer's disease. Most of the people in this are pretty mild because the protocol involves a lot of different types of testing, and people who are more moderate to severe, just it's difficult for them to do. So nobody's actually supposed to be able to see this. I can't even see this. I can't even see it when, on my computer. This is the outline of the ADNI studies as they've rolled over from the different phases and the different phenotypes that they collected each one. The whole purpose of this was to just show you that it's very complicated and there's a lot of moving parts to this study. So I broke it down to hopefully more visible sorts of little breaking points. So ADNI 1 enrolled about 800 participants. At the initial visits, they collect the general sorts of demographics. They also do a medical history exam and physical exam. ADNI 2, or ADNI Go, enrolled 200 EMCI, as I talked about, and ADNI 2 then enrolled another approximately 600 individuals within each of these groups, as well as 100 SMCs, which were tacked on at the end of the study. And then there were people who rolled over. So ultimately, through ADNI, up to ADNI 3, which is just starting, there's been over 1,700 people enrolled. So at the screening visits, and then first of all, so there's follow-up visits either to start every 6 months for ADNI 1. For ADNI Go and 2, they actually threw in a 3-month window for a follow-up visit, and then it's annually after that. So every year, people come back in and get some combination of testing. So the cognitive phenotypes. Mm-hmm.
Sorry, just a quick question. So across the ADNI waves, how diverse in terms of race and ethnicity are these waves?
Not very. I would say it's probably, I want to say it's about 92% non-Hispanic Caucasians. So it's, and that other 8% or so is a mixture primarily of African Americans, but there's some Asians. So then it even breaks into smaller pieces. So it is a largely white, largely non-Hispanic population. And it comes from many sites across the United States and Canada, mostly academic medical centers, but it is not a very diverse population of people, at least in that way.
And while you're talking about that, can you tell us the educational level of the combined samples now?
About approximately college education, so probably 16 years, roughly. It does. There are some people. I think the minimum amount was 8 years of education, a formal education, but I would say on average it's running about 16 years. Mm-hmm.
And what's the recruitment strategy?
It probably varies center to center, would be my guess. A lot of the academic medical centers, they're going to be people who come in a clinical setting and then get sort of offered this as a study. Others would be more, recruitment from the community with flyers and those sorts of things. But it's a very complicated study, and you do multiple types of imaging. It's probably hours and hours and hours. So typically, I would say you have to have people who are very motivated because annually you get scanned and probed and prodded and asked all kind of questions. So.
But I don't think it's motivation that.
Oh no, I don't think so at all. It's I just mean that more in generally. I mean, I think ADNI never made any real attempts to recruit from more underserved populations or from really lower educated. I mean, they're primarily just trying to get people who will do imaging as quickly as possible.
Shannon, wasn't recruitment also based on diagnosis at baseline?
Yes, you had to. Yeah, they were shooting for a certain number. So in ADNI 1, it was 200 controls, 400 MCIs, and 200 ADs approximately. And so depending on what bins they had open, people may or may not have been recruited.
Was there E4 selections
No.
Or not?
But
No.
they didn't recruit, I mean, but there was like some cardiovascular.
Yes, there are medical comorbidities that would exclude people from being recruited. Primarily, these are things that would really impact, like, were not controlled, it would really impact things. Anybody that they really thought had a major stroke or those sorts of things. I mean, they were really wanting to target late onset Alzheimer's disease. Now, that being said, there were a number of people, and Liana knows, that actually probably had an earlier age of onset, less than 65. But primarily, this was supposed to be an Alzheimer's population. So if somebody thought that the dementia was caused by a vascular cause, they likely would not have been enrolled. Okay. So the cognitive phenotypes, which I know is one thing we're going to focus on quite a bit for some of these studies, were done at baseline and then at every visit after that. So whether it be 6 months or annually. And there were a number of different tests done, but a lot of these were very general dementia tests. So there was an AMNART given at baseline to generally get educational or, the sort of underlying IQ. Then the CDR, the FAQ, the GDS, the MPI, ADAS-Cog were all done at every visit. And then there was a neuropsych battery, which I will go show in the next slide. The MoCA and then 2 measures of cognitive complaints were added as part of ADNI 2. So these are only available in ADNI 1 for the people who rolled over, which is kind of a bummer, but it is what it is, for those of us interested in subjective cognitive decline. So the ADNI 1 go-to battery, the battery that was consistent, included the MMSE, logical memory, the RAY, auditory verbal learning test, clock drawing, trail making, animal fluency, and the Boston naming test. ADNI 1 also had a vegetable fluency and digit symbol and digit span. But these were dropped in ADNI 2, and I was not involved in the decision making for that, so I don't know why. But if you're interested in them, there's at least 800 people who have them for a while. And like I said, ADNI Go-To, they added the MoCA and they added the ECOG. And the CCI-20, which was Andy Sakin's baby, was only administered, despite our protestations, to the SMC participants at enrollment to determine whether they were so-called subjective cognitive decline, significant memory concerns. It was not repeated at any time after. Hopefully that'll be different in ADNI 3, but we'll see. So then I'll switch to sort of the other types, the other phenotypes that they collected. So the fluid phenotypes, they do screening labs at baseline just for things like cholesterol, sort of a CBC panel. Plasma and serum, at least as far as in starting in ADNI Go to ADNI 2, are collected at every visit. So there are a number of panels of different types of markers that have been done on a lot of these people. There's also RNA collection in ADNI Go-To phases.
Mm-hmm. So who ran these samples? Different labs, different protocols, or a centralized?
So they all get shipped to a centralized location, which I believe has always been NICRAD, which is the National Cell Repository for Alzheimer's disease. But doing the actual analysis of the samples is done by specific labs. So like specific labs can request samples. So that you can say, I have the greatest new thing on blood biomarkers, known to man, and I want to request ADNI samples to try to test my hypothesis. And so then they would be shipped to that location where they would run the analysis, and then the data would then be uploaded to ADNI site. So then you, as a user, could go and download what they did and get their analyte measures. But those are done at a number of different institutions that can be really all over the world. Okay. So CSF, which at least for ADNI1, is the only real way we're going to get to potentially underlying amyloid and tau pathology, was only done in 50% of the individuals in ADNI1, which is unfortunate. But they realized their mistake, rolling over into ADNI-GO and 2. And at that point, everyone got CSF, nearly everyone. Everyone was supposed to. Got CSF analysis at the baseline visit as well as then every 2 years after that. So looking at the MRI scans, again, it's somewhat complicated, and it's changed over the course of the study. ADNI1 was on 1.5 Tesla magnets. Everybody got that. There was a small subset, a quarter of people who got 3 Tesla scans. But then switching to ADNI-GO and 2, they then switched everyone to 3 Tesla scans and changed the scanner sequences in some ways. So it makes combining the two a bit tricky. So it can be done, but it's difficult to do when you've got different scanner strengths. FDG PET was consistently done across ADNI1, GO, and 2, with 50% of ADNI1 having FDG PET, and then all of ADNI-GO 2 at baseline and then every 2 years. PIB PET was an add-on to ADNI1, so like just a handful of people got it at their baseline visit if they were like the last people to be enrolled in ADNI1. Most of the other people, if they did get it, they got it at a follow-up visit, and only about 100 people got it in total. And ADNI-GO and 2, they did florbetapir on everybody. So they got that at baseline and again every 2 years. And that allows the researchers to pretty much generally be able to tell, at least between CSF or florbetapir, whether people were amyloid positive in ADNI2. Flortaucipir, or AB1451, or whatever you want to call it, is recently been started. It was started maybe a couple of years ago, and the earliest follow-up visit in people from ADNI2 was about 24 months in. Some of the people were already, 10 years in, some ridiculous amount, if they were ADNI1 rollovers. And so that data, there's now approximately, I want to say about 170 people who've had flortaucipir PET, but those have only been very recently. And these are the imaging phenotypes. So that, as I mentioned, they changed the sequences slightly. It's probably not super important because we won't necessarily be dealing with brain images directly. But in the consistent things between ADNI1 and ADNI2 were that they did analysis of brain structure and of white matter damage in different ways. In ADNI2, they added sort of these advanced MRI techniques, things like diffusion tensor imaging to look at white matter integrity and connectivity, ASL to look at brain perfusion, and resting state fMRI to look at functional connectivity, but only about approximately a third of the sample got each because they limited them to a single platform. So certain people on GE platform got one, Philips got another, Siemens got another. So important things to know when you're using the ADNI data set for anybody who hasn't used it before. The RID, it's what they call their unique subject ID. It's different for every person, and most of the phenotypes are coded by this. The patient ID is a full ID that includes both the site number and the subject ID. So that last piece of the full ID is actually the RID. And images and genetics are coded by this ID. The visit code is exactly what it sounds like. It's the visit at which the data was collected. So a lot of the data we're probably using is going to come from something called the ADNI merge data set, which is a merging of a bunch of different sheets together. But that may or may not have everything you're looking for. Depending on what you're interested in, you may or may not want to go above the data that you're given here, to start and play with. So these are some of the sheets, and I won't necessarily read them, but these are some of the sheets that you might find something that you might be interested in, whether it be medical history or specific types of imaging. And these are definitely not anywhere near the number of sheets, just some that I could fit on a slide. So for those of us who work with ADNI a lot, this will be very familiar, but most of the data you'll use will be provided by Paul and everyone else. Very nice to provide. But this is what, if you wanted to go to the ADNI site and download it, this is what it would look like for the clinical data. So this is only like the screenshot that I could get at the top of my screen. It goes way down. It's under, study data. So if you go and you log in, I didn't go through everything, but if you go and log in, everybody should have asked for data access, and they got to log in. You go to the download, and then you go to study data. And study data is going to give you all the phenotype data. And different sheets, and each one of these are CSV files. If you actually wanted to be a go-getter and go actually get some of the imaging scans, you would go to download image collections. And there you can actually search what kind of image you want, what visit you want, other types of search functions. And this is the genetic data, so you go to download genetic data. And this will give you the genotype data as well as some of the more limited files that are associated with they did both whole exome sequencing and whole genome sequencing on subsets of the cohort. So when looking at a data set, I already kind of said this, but this is what would RID, patient ID, looks something like this. And then they do the visit codes by months, so it's like a month follow-up. So like that would be 3 years. It's not rocket science. All right, I will look at my time and see where I'm at. So I just wanted to briefly go over what types of imaging phenotypes there are in case people are interested in analyzing these. For the MRI scans, there's generally a few different ways in which people will go about analyzing these. There's lots of ways, but I just sort of picked a couple. One sort of method is to actually look at it on a voxel-wise basis. So actually look at every small unit of the image and do some sort of statistical analysis on it, whether it be based on shrinkage, whether it be based on tissue characteristics. The other would be to take what we know of anatomy and try to pull out specific information about certain areas that you might be interested in. So these would be segmenting and parcellating the brain up into areas that anatomically make sense. Similar types of things can be done with the PET scans. So the previous one was more structural MRI. You can do it on a voxel-wise basis, or you can select out your favorite region of interest, or ROI, and take the PET signal from that region and say, run a GWAS on it or something like that. So this is a slide courtesy of Andy Sakin. He went through and made this nice and square. Looking at sort of the different sort of levels of granularity that we can have both on the genetic side as well as on the brain imaging side in this case, but in sort of the phenotype side. So you can go from a single region, let's just say the hippocampus because we like that region, and you can look at it either on a single SNP level, like a single gene or SNP that you're interested in. You can look at a biological pathway or do a GWAS. On this direction, you can move from a single region of interest to something more like a brain circuit. Regions that maybe are involved in a resting state network or some other brain circuit that you're interested in. And again, you can do different types of genetic analysis across those. And then you can move to a whole brain, in which case you can either, the easiest would be to start with just one gene, ApoE being the main one. But you can also do it on a pathway and a whole genome type approach. So I'm going to switch and go through just some examples of each of those 9 boxes as best I could from the ADNI study. So I apologize in advance if your study is not on this slide, because there are a lot. As you could start from that beginning, there was a lot of ADNI studies. And so my goal was just trying to pick one that fit in each one of those 9 little squares. So there are lots of them within each one that could have been up on the slide. And please don't take it as a personal insult. And Paul, do you want me to stop, or do you want me to?
You have another 7 minutes before my alarm goes off.
All right.
You're doing
All right.
perfect.
I don't want to miss lunch.
Nobody's going to miss lunch.
No, I promise I'll stop. So the great thing about ADNI really is the wealth of phenotypes we have, but that doesn't mean people haven't done case control analyses. Often ADNI will be, because sample size is king in genetics, ADNI really isn't that huge, despite the fact that in imaging we think it's enormous. So there are some GWASs done for case control, but it's really usually a much larger cohort. But sometimes people will use ADNI as a replication sample. So they'll say, okay, I found this really cool gene in my GWAS. I want to then look in ADNI and see if it also is associated with Alzheimer's risk. So that's what was done. And this paper was looking at whether or not SNPs in the ABCA7 gene were associated with AD risk, and they did find, in fact, it was. You can also do a more pathway or network-based. Sometimes in my slides, it's not going to be exactly that. It's going to be more of like a multi-gene approach. But in this case, they were looking at genes that were involved in oxidative phosphorylation. So they had some hypotheses about why this might be involved in Alzheimer's disease. And so they took their network of genes and made a risk score by combining a risk allele from multiple different SNPs, and found that, in fact, that having a higher number of these risk SNPs in oxidative phosphorylation genes was associated with a higher likelihood of Alzheimer's disease. And then Allison already explained this one. So ADNI was included as part of this massively large thing of 74,000 and some odd people. They were a very small subset of that. So then some studies have been done on clinical status, and I define sort of clinical status not just as case control, but other types of things like the FAQ, which would give you a sense of impacts on ADLs, other types of clinical status, things like depressive symptoms. So looking at single gene, and a ton of times when I show a single gene, it's going to be ApoE. I tried to pick some examples that weren't ApoE, but a lot of times they're going to be ApoE. So in this case, they were looking at whether the E2 allele was protective for functional decline over time. And in fact, they showed that those who carried an E2 allele showed lower functional decline. This is Kwansik No, who I think is still here. Maybe he left because I was boring him. Probably because I said I was going to focus all the questions to him, and he fled the room. So this was looking at depressive symptoms. This included ADNI, but included a couple of other cohorts, and they were doing, he did a pathway-based approach of different SNPs that were involved and had been previously implicated in depression and other types of psychiatric disorders. Then looking at cognition, and I define sort of cognition as more specifically like a memory or an executive function. You can look at single gene, again, ApoE, which in this case was interacting with age to lead to greater decline in this case, language function. Then in this case, it was not exactly a pathway, but it was AD risk genes that Beth Marmino published the impact of a polygenic risk score using the top IGAP SNPs. And showed that a higher number of these risk SNPs was associated with both lower baseline, as well as at least in some cases a trend for change over time. And Vijay Ramanan from our lab did a GWAS on memory performance. This included not just ADNI, but also the health and retirement study, as well as some local data, and found some top SNPs, and Vijay was nice enough to actually put the RS numbers in most cases, and was able to identify a few different pathways and genes that were implicated in memory performance. CSF, there have been single genes, and like I said, there's been a ton of ApoE ones, but I wanted to pick something new and exciting. In this case, SORL1 and its impact, a SNP within SORL1 and its impact on CSF A beta levels. A paper in 2015 looked primarily at immune-related pathway genes, so genes involved in the immune pathway, and actually was looking at some gene-gene interactions, and in this case showed that some SNPs in IL-6R were associated with higher A-beta p-tau ratio. And then there have been a couple of different GWASs of CSF, A-beta, and tau. This is one of the largest ones, at least for the time. And, of course, they have ApoE, but they also had a couple of other hits. Basically, what I was doing is I was going through, as best I could, each sort of phenotype class and going through single gene analyses, more pathway-based, and GWAS types of analyses. So there have been a couple of studies in blood biomarkers. And in this case, they were looking at plasma levels of ApoE, and we're looking at a specific gene that altered the relationship between plasma and CSF levels, one of ApoE and plasma ApoE and CSF A beta. This was a study actually from our lab, which was trying to link proteins in blood samples to their specific coding gene, and looking at the association of how strong different types of genetic variants in those genes predicted blood biomarkers of certain protein levels. MRI has been an area that there's been a lot of studies, both on a single gene level like ApoE, as well as on pathway-based types of analysis. There's some with the immune pathway. I highlighted a study earlier. This one was one with genes related to inositol processing. And then there's actually been both voxel-wise GWASs, so actually on every voxel in the brain, doing GWAS, which, as you can imagine, is a lot of pieces of information in both directions. As well as I'm throwing in a little whole exome sequencing here and highlighting Kwanzigno, who has now rejoined us. And this was an extreme phenotype design. There have been a very limited number of studies in the advanced MRI techniques. I'm highlighting a couple of them. There really aren't that many. So the first was arterial sclerosis genes and their relationship to ASL, which is blood perfusion of the brain.
Yeah, it's hippocampal sclerosis as well.
Yes, that's right. Yeah, it was from your guys's. Yeah, so it was a hippocampal sclerosis and an arterial sclerosis gene. And they were actually using ADNI as sort of confirmation of looking at it in vivo, because it came mostly from autopsy data. And a couple of things have been done in resting state functional connectivity, mostly in the ApoE domain. In this case, this was connectivity, functional connectivity of the left hippocampus. And a couple of studies have been done with the diffusion, but I've obviously selected one sort of for each of these three different techniques, but they're very limited. So if I know we're working mostly on ADNI 1 in this particular conference, but if you were interested in this is an area that, especially rolling into ADNI 3, which is where everybody will get one of each of these types of techniques, might be a really interesting thing to do for GWAS and other types of genetic analyses. Moving to PET, there are quite a few studies of amyloid PET. In this case, this was a study of ApoE, linking ApoE to a great deal more amyloid, regardless of your clinical status. There's also a pathway based in the immune pathway, looking at the interaction of two different genes on increased amount of amyloid. And a couple of GWAS studies, one on cross-sectional amyloid, and this one was a Manhattan plot of longitudinal change. There's also been a couple of studies on FDG PET, which are actually more limited than I would have expected, but going back through this literature, there really weren't that many that looked at GWAS or anything, or even pathway-based. Most of it was single gene. So this is examples of ApoE effects on FDG metabolism and AD risk genes. In this case, I'm highlighting a result with CR1, but there was a panel of 20 AD risk genes. So for more information, see these two reviews, one from Andy about the genetics core of ADNI, and the other was, I borrowed on heavily from Li Shen from our group, essentially explained all of these different types of genetic and imaging and cognitive analyses in a number of different domains. So I hope I've convinced you that ADNI has a number of longitudinal phenotypes to test not only genetics, which is our focus here, but any number of hypotheses that you might have about Alzheimer's disease. A number of them have been, genetic studies have been performed to date, but there's still a lot of missing sort of gaps, particularly in the advanced MRI, and most recently tau PET being added. There, so far, have not been any genetic analyses of the tau PET reported, and there's more data coming. And this was the shameless plug that was Andy sent me and said, please put this at the beginning of talk. And so please come see us in Indiana. It's really a lovely state, and we don't all race cars, just some of us. Just some of us.